How's it going, you guys? So in this video, I wanted to discuss phylogeny. So phylogeny is considering the evolution of a species or a group of species, but really considering their history and what led them to evolve to get to this moment that we have. So it also kind of considers the relatedness of species and their historical divergence, so to speak. So that's phylogeny. It's the evolutionary history of a species or group of species. Now, when we think about phylogeny, we really consider it as a branch diagram, if you will, and it really comes in two forms. One is a phylogenetic tree, which is a true evolutionary tree, and the other is a cladogram. So we'll talk about both in a second, but I want you to have some terms down that we use for both cladograms and phylogenetic trees. So this is a picture that I tried drawing that resembles an image in your notes. So I would say find this image in your notes and then mark it up as I do on the board. But here is a tree that shows the relatedness between several species. So we have species circle, species triangle, square, diamond, hexagon, and then smiley face. Now here, all these species are related ultimately together, and they share a common ancestor, but some might be more closely related than others. So that's what a phylogenetic tree or cladogram seeks to answer. What is the relatedness between species? Now, a few terms that you should know. This common ancestor down here, so we see that all of them ultimately branch from a common ancestor. So this common ancestor is considered our root. So we consider that the root. All of these other points where we also have some common ancestors, here, 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 and here, those would be considered nodes. Or internal nodes. Because we also have terminal nodes, which are these guys right here, which are the final species. So boom, boom, those would be terminal nodes or tips. So we consider these the tips or terminal nodes. These are just regular nodes or internal nodes. And this is our root, our oldest common ancestor for all of the species depicted. So those are some terms that you should know. This right here, a single line on this diagram, whether this or this or this, that would be considered a branch. So a branch is a line on a phylogenetic tree or cladogram, and a branch ultimately shows the history or the time between the common ancestor and the modern day species. Now, for this particular phylogenetic tree or cladogram, it's important to note that the branch, it might be not including certain organisms, certain other divergent branches. So we could see a branch here, another branch here, but we're just not depicting that in this particular tree. So they could be there, but we're just not showing it. This right here, if we consider this tree, this guy right here would be our outgroup. So that's our outgroup. And we know he or she, or that species, is our outgroup because it diverges from the oldest ancestor, but it doesn't have as similar relatedness to all these other species depicted. So like this species and this species have a more common or more recent ancestor than with this species. Same with these two species. Same with this and this species, right? They have a common ancestor that is earlier in history than the common ancestor with the outgroup. So this whole group right here would be our in-group. So we have our outgroup, we have our in-group. If you have two species like this that share a fairly recent common ancestor, we would consider those to be sister taxa. Sister taxa. And that's about it for the terms that we need to know for, or the initial terms, I should say, that we need to know for phylogenetic trees or cladograms. Now, I also want to point out that I drew it like this, but we very well could have easily flipped these two, and this would still be considered accurate. So this would still be the same if we just flipped this and had this hexagon species in this spot, and this diamond species in this spot, right? So if we just switch the terminal nodes or the tips, as long as we keep the branch order the same, that's fine. We could have also switched this entire one. We could have rotated it on this node and switched this entire branch. We could have rotated it there, rotated it there, rotated this here, and it would still be considered accurate. So the order of the tips, the order of the terminal nodes doesn't really matter. What does matter is the topology. The topology is the order of the branches. So this branch pattern needs to stay the same, however we flip it. Also, we could have very well drawn this not like this. So we drew it in a very specific way where we have kind of like a V. We could have also drawn it like this, where it branched off like that and then branched off like that. That would have also been fine. But again, we need to make sure that we have our topology correct. The topology doesn't change. So that is that. Those are those. This includes some of the initial terms that you need to know. Now, continuing on with phylogeny, I want to talk about phylogenetic trees versus cladograms. So they're very similar with just a subtle difference. So phylogenetic trees are evolutionary trees. They are true evolutionary trees because they don't just consider the relatedness of the species and their common ancestors, but it also considers time. So not just the fact that species A and species B share a common ancestor, but the fact that they share a common ancestor maybe exactly 1 million years ago. So we consider time when we talk about phylogenetic trees, whereas cladograms, we don't really consider time. We just consider their relatedness. So if you look at them side by side. So here's a phylogenetic tree, and here is a cladogram. Let me adjust this slightly. So phylogenetic tree versus cladogram. What I've done is taken the liberty to draw in 1 million years ago, 2 million years ago, 3 million years ago, and 4 million years ago, because phylogenetic trees consider time, right? And so you can see the length of their branches also are drawn in conjunction or considering time as a factor. So if you look at this divergence right here, after species G, once there's a divergence here, you see that this branch is shorter than this branch. That means that this divergence right here happened not as long after this divergence when compared to this one. So this was a shorter amount of time than this amount of time. So hopefully you can all see that. Even if we didn't draw in 4 million years ago, 3 million years ago, 2 million years ago, or 1 million years ago, even if we just left it like this, we could still see that this is a phylogenetic tree and not a cladogram because of the differences in branch lengths. And because we are considering the fact that the branches are different in size as a relative factor to time. We also see here that we see species G, species E, species B. All of them are not at modern time because G, E, and B have all gone extinct. Maybe just over 1 million years ago, just over 2 million years ago, between 3 and 4 million years ago, species G went extinct. So phylogenetic trees consider time, whereas cladograms, as you can see, they're not considering time. So all of these branches are relatively consistent with their distancing. So there's no consideration of how long ago this divergence occurred or this divergence occurred, just the fact that it occurred. And we see the relatedness just as we see that in phylogenetic trees, but again, no time. So that's the big difference between cladograms and phylogenetic trees. They're very similar in many ways. Phylogenetic trees tend to be more reliable because they are also considering time. But it's important to note that even though phylogenetic trees are maybe more reliable than cladograms, they're both really considered hypotheses. So while they're supported with evidence, they might be changed based on the introduction of new evidence. which we'll talk about later on in a little bit more depth. But what is that evidence that we use to determine the relatedness between species? Well, we ultimately use data. We ultimately use data from two sources. We look at morphological data, so how are they similar in their anatomy, their bodily structures, and how are they similar at a molecular level, so their molecular data. So we look at the proteins that they share in their cells. We look at their DNA, the similarities between their DNA to determine how closely related they are. And based on morphological and molecular data, we can ultimately put together a pretty decent cladogram or phylogenetic tree. Now, it's also important to note that when you're trying to assemble a phylogenetic tree or a cladogram, yes, you have to consider the morphological differences and the molecular differences. And let's say we see this group that has hooves, like species A and B have hooves, and species C doesn't have hooves, chances are these two are more closely related than this one, but that's not always the case. And it's also important to note that we can sometimes just as easily lose traits as we can gain them. So for instance, let's say we have a species that has spikes, it has, let's say, eyes, maybe it has hooves, maybe it has a tail, and maybe it has sharp nails, right? So those are 5 traits. Now, if you see another organism that has all of those 5 traits, but maybe is missing eyes, maybe is missing one, chances are it probably diverged from this one organism that had 5 traits and diverged with a common ancestor with that one than it did to come up with all 4 traits on its own without eyes. So that idea that we go with whatever explanation is the simplest, where it's easier to lose one than to gain 5 traits throughout history, and be that similar, that is Occam's razor, this preference for simplicity. Whichever is the least complex explanation is probably correct. So we'll talk more about that. You'll probably have a chance to design your own cladograms in the not too distant future. But one other thing that I want you to understand, some other terms that you should know before we wrap this idea up of phylogeny, is some other terms such as monophyletic group, paraphyletic group, or polyphyletic group, as well as synapomorphy. So a monophyletic group can also be called a clade. We'll talk about what this is in a second, but mono, if you guys remember, means one. Para means near, and it takes on a couple different meanings, but I'll explain para meaning near and its significance here. And then poly means many. So if we look at this hypothetical tree that we have here, this hypothetical maybe cladogram, we might see, so let me erase this real quick. We might see that we have species A, B, C, D, E, F, and G. If we consider species E, F, and G, we would consider that a monophyletic group, or a clade. A monophyletic group is a group that has one common ancestor and all of that ancestor's descendants. So E, F, and G are all descendants from this common ancestor right here. So this, in orange, is a monophyletic group. Now, let's say we considered species D, E, F, and G, and we grouped them together. That would no longer be a monophyletic group, because their common ancestor, their most recent common ancestor is right here. This common ancestor has G as a descendant, F as a descendant, E and D as descendants, but it also includes B and C as descendants. So because we're not considering B and C in this group in green, we consider that a paraphyletic group. It's near or almost a monophyletic group, but it's leaving some species out. So that's a paraphyletic group. Now a polyphyletic group, if we considered species B, E, and G, so now we're just randomly, almost randomly, sampling from the species in consideration without actually considering their common ancestor. So E and G have a common ancestor here, but we're not considering F. B has a common ancestor here with E and G, but we're not considering C, D, or F. So if we just consider species without their common ancestor, so we're randomly sampling them, almost randomly sampling them from a tree, then that is a polyphyletic group. Poly meaning many, so we have many different spots in the tree. And again, A would probably be our outgroup, or is our outgroup, and B, C, D, E, F, and G would be our in-group. So those are some terms that you should know as well. Monophyletic group, or clade, paraphyletic group, and polyphyletic group. The other term that I had over here was synapomorphy. So a synapomorphy, let me bring you back over here. A synapomorphy is a characteristic that is found in an ancestor that that it has been modified, I should say, over the course of evolutionary history, and is found in all of the descendants, but may be modified slightly. So, for instance, if we have a characteristic, and then over time, species diverge, we might see morphological homologies, like the one bone, two bones, many bones, that we saw, or that we discussed with the Evidence for Evolution Part 1 video. But these morphological homologies ultimately derive from a common ancestor's synapomorphy, that common trait. So here, if we consider a synapomorphy in this tree, okay, so if we consider this synapomorphy being the first organism that had the one bone, two bones, many bones, so if we consider the first organism that had the one, two, many bones trait in the limbs, that has ultimately diverged to maybe fins, to maybe wings, to maybe arms or legs. So ultimately, synapomorphies is the common trait, or the trait found in the common ancestor that has diverged into morphological homologous structures. Now we're almost done. We have one last thing to talk about, and I mentioned this earlier. I said I would come back to it. And that has to do with the fact that phylogenetic trees and cladograms, they both are hypotheses, right? So it's possible that they could change. A good example of this was whales, hippos, and pigs, right? So if you looked at a whale and a hippo and a pig, you'd probably say that the hippo and the pig are more closely related than the whale and the hippo, right? But it turns out that was based on morphological evidence, but it turns out after looking at genetic evidence that in reality, the tree looks more like this. So hippos are actually more closely related to whales than they are to pigs. So that might surprise you, but if you look at the genetics, if you consider the history, whales at one point were four-legged creatures that walked on land that were entirely terrestrial, did not live in water, and gradually became more adapted to living in water. So even though you might look at hippos and pigs and say they both have four legs, they're more closely related in size than the whale, if we look at the genetic evidence, the molecular evidence, hippos, it turns out, are more closely related to whales than they are to pigs. And so analogous structures also, if we consider this, can be misleading. If we see wings, for instance, we talked about analogous structures as an example, like wings. If wings develop in a couple different ways, that doesn't necessarily mean they're very closely related, that we should group all of the organisms that have wings, because they might have developed through convergent evolution as opposed to divergent evolution. Convergent meaning that they separately led to the same general characteristic because it's evolutionarily beneficial. So I hope that video was helpful, and if you have any questions, leave them in the comments, send me an email, and let me know.